Okay, so question 7 is a reading comprehension question. So I'm going to give you guys some time to read on your own. You guys can pause the video if you want, but I'm just going to go ahead and answer the questions. All right, so A1, it says, free radicals can damage genetic material in the cells. Describe one effect that this could have on the body cells. So if it's genetic material, we're talking about DNA here. And if you're damaging the DNA, then that could lead to mutations, which could eventually lead to cancer, for example. So this can cause gene mutation, which would lead to cancer. You could also say that because genes basically give you the instructions on how to make proteins, if you have a mutation on a gene, then that would affect how the protein is being made. So that could change the shape of the protein or change the function of the protein. And part 2 suggests how antioxidants such as vitamin C allows cells to function normally. So we need to look at lines 5 to 6 and 8 to 10. So 5 to 6 and 8 to 10. Okay, so we can see in lines 4, 5, and 6 that these free radicals can affect the flow of substances that leave and enter the cell. And in lines 8, 9, 10, vitamin C is known as an antioxidant. Antioxidants protect cells from damage by free radicals by changing the way the free radicals behave. So basically what we would say here is vitamin C is an antioxidant. It changes how free radicals behave. Therefore, it can prevent from affecting the transport of substances inside and out, in and out of the cell, of substances in and out of the cell. Okay, part 3, calculate the mass in grams of boiled broccoli that should be eaten to provide the recommended daily intake of vitamin C. So this is asking for boiled broccoli. So it says here for 40 grams, for example, 40 grams of fresh broccoli provide 80%, but the same mass of boiled broccoli provides 60%. So if we have 40 grams, that would give us 60%. If we want to get 100%, what should we do to this number? So to get this from 60 to 100, we need to times this by, well, first we need to divide it by 60 times by 100. And we would do the same thing on this side. We would times that by 100 divided by 60. So 40 divided by 60 times 100, that would give us 66.67 grams. So we can write that as 66.7 grams. All right, DCPIP is a chemical used to determine the amount of vitamin C in food. Describe a method using DCPIP to compare the amount of vitamin C in fresh orange juice with that in boiled orange juice. So in the previous, I think in the last paragraph, it talks about how boiling is the biggest cause of vitamin C loss, right? So there would be less vitamin C in boiled orange juice. So DCPIP is a reagent that is blue in color. So I'm just going to write this down. So DCPIP is originally blue, but in the presence of vitamin C, it actually becomes colorless. So what we can do is we can have, let's say, a known sample of fresh orange juice. So let's say 5 cm cube of fresh orange juice and 5 cm cube of boiled orange juice, for example. And we would use a burette to add drop by drop of DCPIP to both the fresh orange juice and the boiled orange juice and compare the volume of DCPIP added to these juices. So let's write this down. Pour, let's say, 5 cm cube of fresh and boiled orange juice into separate test tubes, and then we would use a burette. Add drop by drop of to the juice until it no longer decolorizes. Because what happens is DCPIP is blue in color, and in the presence of vitamin C, it becomes colorless. Then we would record the volume of DCPIP added and compare the results. Alright, so we get 1 mark for adding the fresh and the boiled orange juice into a test tube. That's 1 mark there. We get 1 mark for making sure that we use the same volume. We get another mark for adding a drop by drop of DCPIP. That's 3 marks. Record the volume. That's a 4th mark. And compare the results. That's the 5th mark. Okay, let's go on to question 8. Okay, Duchenne muscular dystrophy is a sex-linked genetic disorder caused by a gene mutation. The symptoms of DMD are caused by the absence of a protein called dystrophin. Explain how a gene mutation can cause the absence of dystrophin. So I've included a little diagram here to explain to you how the gene mutation could affect the protein. So here we have a normal DNA sequence, right? And that DNA sequence would lead to the formation of the mRNA, and that would lead to, so each of these 3 letters represent a codon, and that would correspond to a specific amino acid. Now, if we were to have a mutation, so in this case, instead of G, there's an A, that would lead to a new codon that gets formed, which means a different amino acid is added on, except instead of histamine, we would have tyrosine. So that's one way in which a gene mutation could affect the protein synthesis. Another way is there could be a stop codon. So a stop codon prevents the formation of the full protein. So for example, if your protein has 100 amino acids, a stop codon, let's say, can cause the amino acid sequence, can cause the protein to form up to, let's say, 36 amino acids. So instead of your protein being 100 amino acids long, it will be like 36 amino acids long, which is what we refer to as truncated. So let me just make this one smaller and add this, write this down. So, okay. A change in the base sequence could produce a different mRNA. This would lead to a different codon and therefore a different amino acid. And then we could also say a gene mutation can cause a stop codon. So also lead to, whoops, it could also produce a stop codon, lead to a faulty protein. So here we've got change in the base sequence, we have different codon, therefore a different amino acid, and then I've included producing a stop codon, which could lead to a faulty protein. That is 1, 2, 3, 4 marks. Okay, so part B. The diagram shows the inheritance of Duchenne muscular dystrophy in one family, and here's a pedigree. Explain what is meant by a carrier of DMD. So essentially, a carrier is when a person is heterozygous for the recessive allele that causes the disease. So they present as healthy, but they do carry the faulty allele. So here we would write, so this is when a person carries the faulty allele on one of the chromosomes, one of so the dominant allele would mask the effect of the recessive allele. The dominant the effect of the recessive allele. Alright, moving on to part 2. Explain how the diagram shows that DMD is caused by a recessive allele. So if we scroll back up, right, they mentioned earlier here that DMD is a sex-linked genetic disorder, and so that means that for most of the time, the faulty allele will be on the X chromosome. And so because females have XX chromosomes, then they can be a carrier. So for example, this parent here, this is the mother. So the mother has, or the mom has, 2 X chromosomes, and the faulty allele, let's assign that as lowercase d. So the mom would have 1 dominant allele and 1 recessive allele, right? And this father here would be XY, and because the father does not have DMD, that means that he carries the dominant allele. Now, their son, which is this person here, is the only one that has DMD. And so, well, we know the son must inherit the Y chromosome from the dad. So let me just write that there. So the Y chromosome must come from the dad, which means the recessive allele must come from the mom. Right, so that's there. So if we scroll back down, we could say, well, the mother. So females have the XX chromosome. And the mother, who is the carrier, carries the faulty allele on one of the X chromosomes. One of her X chromosomes. Her son, so in generation 1, let me just double check that. Yes, so the son is in generation 1, right? In generation 1 has the XY chromosomes. And inherited the faulty allele from his mom or his mother. Okay, let's move on to part 3. Draw a genetic diagram to show the inheritance of DMD from the parents to generation 1. Okay, so we've already wrote the genotypes here. So this is the genotype for the mom, this is the genotype for the dad. So let's draw out the Punnett square. So we've got the mother's gametes. So that's XD, XD. And then we have the father's gametes. So that is XD. Oh, sorry, that should be a dominant allele because he is healthy. And this is what we would get. Okay, let's move on to the 9th question and the final one. Part A, hepatitis is a disease of the liver. This disease can be caused by drinking too much alcohol. Explain the damaging effects of alcohol on the liver. So alcohol could cause damage to our liver. It can cause things like scarring or cirrhosis. It can affect the functions of the liver. So for example, our liver is responsible for making bile and for detoxifying substances in our body. And the tissue damage that is done to our liver can cause mutations to occur. And with mutations, that would increase the person's risk of getting cancer. So because we only need 3 points for this, let me just write that down. So alcohol can cause cirrhosis, which is the scarring, and can lead to gene mutations, which could increase the chances of getting cancer. So that's 1 mark here, 1 mark here, another mark here, so that's 3. You can also say it could affect the liver's function in terms of detoxifying substances and also making bile, so that's another mark that you can also say. All right, let's move on to part B. Hepatitis B is one type of virus that can cause hepatitis. Describe the reproduction of a hepatitis B virus. So let me just pull up the diagram that we have here. And let's go over this one together. Okay, so here's the life cycle of the virus. So you can see here this virus has attached itself to the host cell, and then it would inject its genetic material. So here it could be DNA
it's coming inside to the host cell. And that DNA can be incorporated into the genome of the host cell. And once that happens, you can have protein synthesis that occurs, and it would use the viral DNA that is inside to create more viral particles. And when that happens, you create this whole army of viruses inside of the cell, and that would cause the cell to sort of burst, or we call that lysis. And that would release the viral particles so that they can infect other cells as well. Okay, let's close that. Let's make this one smaller so we can write this down. Okay, first, the virus attaches itself onto the host cell to inject its DNA inside. Inside. This viral DNA can be incorporated into the host genome. And so new viruses can be produced via protein synthesis that would cause the cell to undergo lysis. So that's when it bursts, right? To release the new viruses, new viruses. So let's go through the points here. We got the virus attaches itself onto the host cell, that's 1 mark, and inject its DNA inside, that's 2 marks. The viral DNA can be incorporated into the host genome, that's a 3rd mark. New viruses can be produced, 4th mark, via protein synthesis, that's a 5th. Undergo lysis, that's a 6th mark. To release the new viruses, that's the 1, 2, 3, 4, 5, 6, 7th mark. Okay, and lastly, let's go on to the last question, part C. Okay, vaccinations can be used to prevent the spread of viruses. Explain how vaccinations can help people become immune to hepatitis. Okay, so when viruses enter our body, what could happen is that it could activate these lymphocytes, and lymphocytes are just one of the white blood cells that we have. So lymphocytes are responsible for producing antibodies, and these antibodies bind specifically to the virus particles, right? So it will bind to it and it will therefore inactivate the viruses. At the same time, you also have memory cells, and these memory cells will stay in our body so that the next time the virus comes along, the memory cells are like, oh, hey, you've been here before, and they're able to undergo mitosis and be able to produce antibodies much faster. So if we have a little graph, let's say, so the first exposure to the viruses, you might have a little bit of antibodies that get produced, whereas the second time around, it's a lot more faster and a lot more antibodies get produced, right? So let's write that down. I'm going to make this one smaller. And then I'm just going to erase this little marking there. So vaccines contain antigen. So antigens are basically found in the surface of either the viruses or any sort of pathogen that would activate the immune system, right? So vaccines contain antigens which are injected and causes lymphocytes to produce antibodies against it. And the reason why vaccines are effective is because of memory cells. So the same time memory cells are produced so that the next time the antigen is present, antibodies are produced more quickly. Okay, we're done with this paper.